Good morning, everyone. I'm very pleased to present you today my virtual talk on nanomedicines that will revolutionize the healthcare. I'm really sorry that I can't be there in person, but I'm sure you are having a great time, and I would like first to thank the organizers for the opportunity to show our work here that we do in Helsinki, Finland. About this topic. So a great pleasure and a great honor to present this to all of you, and I'm really sure that you are having a great time and great talks as well during the past day and this day and next days. So let me just start to introduce you what we do in our lab, in our research group. And first of all, I will just to let you know that we work with nanoparticles, nanomedicines. And why we are interested in this? Well, you all know that the process for drug development takes quite long, many years, and also the investment is quite huge from the pharmaceutical industry and labs. And we initially go from, let's say, 10,000 compounds, then if you are lucky, we end up with one drug that is approved. This is because some of the drugs that are synthesized, they have properties that are difficult to formulate, and therefore we need to develop different systems, alternatives in order to improve their properties. So how we can do this? We can do this by, for example, engineering intelligent therapeutical systems. And in this slide here I showed you a video, but I showed you also here this Fantastic Voyage. So how many of you have seen this movie, The Fantastic Voyage, from the 60s? Oh, if you have seen it before, that's really great. So basically this is what we do in reality. This is what was done in the science fiction movie. So in science fiction movie, basically humans go inside of a patient and they try to correct some of the tissues and cells. Of course we cannot do this, humans cannot go inside of other humans yet, but anyway the idea is that we can micronize or nanosize devices in order to inject them into humans. So this cannot be done, of course, but this one is already a reality. Particles can be injected into bloodstream in the patients, and they can, with proper engineering, they can travel around the bloodstream, find the right place, they can be uptaken by the cells, and then if they carry drugs, they can release the drugs. And of course, for example, if this would be a cancer cell, they can then release the drugs inside of the cancer cell and hopefully kill the cancer cell. So the nanotechnology can be used to help, of course, the medicine. And this has been already envisioned a long time ago by Paul Helrich, who was the first to coin the magic bullet. Basically, we could target specific drugs to specific sites. And why this is important, or why we use nanomaterials, because they have specific benefits. They are a small size, they can have a high density, they can increase the biodistribution in the body, and they can be used for sensing, multi-application sensing, imaging therapy, and so on. This has been very important for pharmaceutical applications. So drug delivery systems have been a huge investment by the companies, as you can see in here, and it is estimated this year that it is even increased to up to $200 billion. And the global market of nanoparticles and nanomaterials are indeed quite high, and you can see here that around $80 billion dollars have been estimated for the next year. So that's a huge market and huge applications. Of course, there are different kind of systems that can be used, from liposomes, the most known, and other inorganic materials and organic materials. So in our lab we are interested in certain kind of biomaterials that I will talk later on. So this is the applications of the nanomaterials and nanoparticles. So it's a reality. There is a huge different kind of applications. Of course, in my lab we are interested in the biomedical and healthcare applications. Why? Because we think that nanotechnology and nanomedicines can help to re-engineering us. What does it mean? It means that it can help to correct some of the disease, it can help to regenerate some of the tissues and cells, and this is obviously a great thing in terms of biomedical applications and for healthcare system in the future. So we are particularly interested on novel nanosystems, biomaterials that can be applied for all kind of biomedical applications. So my group is mainly interested on target cancer immunotherapy, also diabetes and obesity, cardiovascular disease. We are able to make drug nanocrystals by using a special technique we call microfluidics. In Finland we have a lot of different kind of, we have a lot of forest trees. We can extract cellulose and lignin from those and use these materials for biomedical applications. And of course, we can also use them for diagnostics and image, very important, and other different applications, wound healing, spinal cord, antibacterial. And recently we've been quite interested to use these materials, biomaterials, for scaffolds for bone and tissue regeneration. But today I'm going to focus mainly on cancer and cardiovascular disease, and on this particular biomaterial that I call porosilicon materials, and I will come back to that a bit later to discuss about that. So this has been several of applications that we have had throughout the years. These are some examples of the covers that we have published in many journals. So since the scale-up production of nanoparticles and nanobioreactors that we have done, immunotherapy, diabetes, cardiovascular applications, and many other applications. So, but unfortunately I don't have time to tell you our amazing work that we have done during the years, but I will just focus on some examples. So let me tell you why we are interested in this. So more and more we are applying nanomaterials or nanomedicines for different biomedical applications, and in order to achieve what we call a personalized medicine. So what does it mean? We can take all the information of a particular patient, and by doing so we know exactly what kind of drug we should give, at what kind of disease we should tackle, and at what time and with the right doses. This is very, very important. And why? Because target nanomedicines are much more efficient and much more specific than the traditional drugs. So you can see the traditional drugs, they affect the whole human body, but nanoparticles can of course be more specific and target to specific cells and organs. So besides the targeting, we can improve the solubility of different molecules, we can co-deliver different molecules, and more importantly, we can also lower the drug toxicity or the side effects of these molecules for the patients. So by combining nanotechnology, imaging, therapy, we form a field of research, or we are working in a field of research that is called nanotheranostics. So this is some examples of what I'm giving you today. So recently we have reviewed that, of course, there are many different applications and, for cancer nanomedicine, and we need to engineer very precisely the nanoparticles with a certain kind of surface chemistry and different properties, in order so that they can reach to the tumor, cancer tumor, and be delivered the drugs inside of the tumor. And unfortunately, in the past years, there has been not very good communication between engineers, pharmacists, medical doctors. So we have to bridge the knowledge, and more this has been improved, and more we are getting further and better results for the clinical trials. But one of the important aspects is also to really choose the right population that we need to use these materials. Why? Because by doing so, then we can, of course, also tackle better the disease, and we also are really sure that these materials are more efficient. So recently we have just reviewed this, and we know that the knowledge and the BRING is very important, because this is a truly multidisciplinary, multi-interactive research system. So let me introduce you now next some of our current research with the different kind of materials. For example, an example of a biomaterial which is called lignin-based nanoparticles, where we can use for drug, but also for magnetic guiding therapy. So you can see here that we are moving the magnetic particles around, and then we can use them for creating different engineering nanoparticles with different properties, and then we can also use it for anti-cancer applications. In another example, we have this what we call hollow site nanotubes, where you can load different kind of drug molecules. We can design this and load different drugs at the same time inside by using a technique that we call microfluidic system, and by doing so we also can have different drug molecules for anti-cancer purpose, where we can also apply for colon cancer, for example. So how we do these systems? Basically, we can start from a silicon wafer. We can produce electrochemically, by electrochemical etching, different properties of these materials with different porosity. And when we do so, we can also obtain the porous material, the freestanding porous silicon film, and then we can create porous particles, microparticles, nanoparticles. We can make pills and or use them, for example, for imaging. So there are different applications of these materials, from the wafer to powders to tablets and to nanosuspensions. So this is one of aspects by engineering these materials. So we have mastered the application of these materials by doing them with different properties. Then we can, of course, also make them responsive to the pH, for example, and after we give them to cells, cancer cells, they are able to go inside of the cells, be uptake of the cells, release the drugs inside of the cells and kill the cancer cells. And recently we've been also interested to target them to the brain and also to the heart, and I will give you some examples in the next slides. So first let me introduce you some what we have done recently. So we create what we call biomimetic engineering or cancer cell membrane to design nanoreactors with a certain kind of organelle-like functions. So what we did is that we took the cell membrane of cancer cells, we could incorporate porous silicon inside of these, and then by giving them back to the cells, when they go inside, they act as a nanoreactors, where a reaction goes inside, and we can clear up the reactive oxygen species that are produced inside of the cells. So this is very important, because then we can use them to distress the cells somehow, and we can very efficiently see that they are uptaken by the cells, they go inside, and then here we see that the enzymatic reaction takes place and the reactive oxygen species is then decreased. In another study, what we did is that we evaluate that we can target these nanovectors actually to the brain of intratumoral injections, intracranial injections of the particles target. They've been targeted to the neurostem cells, and when they are targeted to the neurostem cells, and by releasing a drug, we have followed what's happened to these cells, and we have been we observed that they were able to differentiate from neurostem cells to actually neurons. And this is a very amazing thing that we observed. And specifically, they only attach and they are targeted to these particular cells. And recently, we also observed that if we inject these particles to the mother with the embryo, the particles are able to travel after the injection to the bloodstream, cross the placenta, and go to the brain of the embryo here. As you can see, these are the embryo of the brain of the embryo, and the particles can be seen here, and we can even follow them by two-photon technique, imaging technique, where we can follow how do they happen. And because these particles are biodegradable, they can, of course, then disappear. They do the job and then disappear. So another way to do that, we can make them multifunctional. So we can do them for theranostics, so it means that for imaging and therapy at the same time. So in this case, we did it for liver regeneration and acute liver failure. So again, we use a technique, microfluidics technique, similar as the one that is represented here, where we incorporate porous silicon particles inside of a polymer layer, acetylate dextran, and loaded with gold nanoparticles and also a drug. This drug is known to be quite toxic to the heart, and it's difficult to formulate. But when we incorporate in our nanosystems, what we observe is that the heart tissue is quite clean, nothing happens, but you can see with the free drug that something happens there. So this is quite toxic. And then when we target them, or when they go inside of the liver, we can see that the liver with the free drug and the drug that is loaded inside of our systems has a much better behavior, and it gives a much better response as compared to the free drug. And besides, the liver is not toxic to the liver, so it does just the job. And we can follow by, for example, imaging CT. We can see how actually the particles are located. So this is another way how to do it. And you can see here what happens. So the drug is the pure drug is extremely poorly water soluble. But when loaded inside of nanosystems, the drug can dissolve actually quite fast. We can increase the solubility of the drug. So this is also very important for the applicability of the material. And again, if we just do some of the in vivo imaging with CT and so on, we can clearly see where in the tissue, in the liver tissue, where the particles are accumulating, and we can visualize them. And obviously, if we are doing some therapy, what we observe is that the particles accumulate and they were able to induce some regeneration of the liver tissue. So this is very important for therapy applications. Now, let me go back to cancer. We know that cancer has a lot of problems. There are a lot of issues that we need to overcome: late diagnostic, side effects of drugs, multi-drug resistance, inefficiency of many nanosystems that we have observed in the literature. So there is something that we can still improve. So and there are many drugs that have been already approved for clinical trials, for therapy, and also for imaging. And so recently, what we have been doing, we have created dopamine-inspired multifunction material where we actually have iron inside of these porous materials, and when we do so, we have a new system here that we call FHP, and this new system here that is shown in this can actually go inside of the cells, can release its pH-dependent release of the drug molecules, in this case doxorubicin, and we were able to demonstrate that we can reduce the multi-drug resistance in breast cancer cells. So this is another application and important of our nanosystems. Together with our collaborators, what we did is that we modified the surface of porous material with this molecule here, and this molecule has a double functionality. So it can be targeted to the carbonic anhydrase 9 of cancer cells that they are in hypoxic conditions, and they also can be used for imaging at the same time. So here you can see the mapping, so we can see how the particles look like. The stability of the particles, they are extremely stable in plasma. And then when we put in contact with the cells, what we see is that in hypoxia there is a huge targeting of an uptake of the particles as compared to normal cells. And more importantly, when they release doxorubicin, anti-cancer drug, we were able to kill the breast cancer cells. So another nice application of these materials. We can make them more complex but still more effective. So we have modified our porous material with different polymers, PEI, and also aptamer, DNA aptamer, and we are able to engineer and modify by using different ratio between the nanoparticles and DNA, the surface charge of these materials. And when we give them to the cells, so here this is very busy, but according to the time and the uptake, what we see here is a large uptake. You can see from this column here, large uptake of our particles, and inhibition, growth of anti-cancer inhibition of the cancer cells after they are the drug is released. So with these systems, we can very specifically target them to different kind of cells. For example, here we show that the previous particles, they do not target nuclear negative cells like T3 fibroblasts, but they highly target nuclear positive cells like breast cancer cells, where they can target the receptors and then go inside and release the drugs inside. So what I showed you was all in vitro, so let's have a look to the in vivo applications. So the in vivo, what we have been demonstrated that these nanovectors, they can target very efficiently into breast and prostate xenografts. For example, we have modified these porous materials with a peptide, targeted peptide, and here you see a huge accumulation of the particles in the breast cancer tissue and no major accumulation in organs like kidney, lungs, heart and brain. And of course all the nanomedicines problem, they accumulate still in the liver and spleen, but still they were non-toxic after we do all the histology, so no problems for those organs. And when we make a multifunctional system where we have a porous silicon particle modified with a targeted peptide, radio label for imaging like SPECT CT, and also fluorescent label, you can see here that after we inject them to prostate cancer, we can see accumulation of the particles there, and they continue for even more than 26 hours, where we can constantly release the drug. In this case, we use anti-cancer drug for sorafenib, and this is what happened. You can see, you can appreciate the survival of animals, the tumor increase. You can see that the tumor volume increases with the pure drug, but when we have our formulation, the porous silicon with the drug loaded and the target, you can see the tumor does not grow so efficiently. So this is another way how we can apply these nanomedicines for cancer therapy. So recently we have been interested to actually apply this also for immunotherapy as a nanovaccines. So what we did is that we extracted the cell membrane of cancer cells and we incorporate porous materials inside of this cancer cell membrane cancer cells. This is what you see. It's a cancer cell membrane, and inside we have actually incorporated porous material. And then we have evaluated all the co-stimulation signals as well as the cytokine release in different cells, PBMCs and BDCMs. And what we observed is that they were able to actually induce some stimulation and also cytokine release. So we were really very surprised and very happy to observe this. And here we didn't use actually any adjuvant. So we use all the properties of the particles themselves. So and then when we actually gave them when we use them for the treatment of aggressive melanoma with checkpoint inhibitors, what we observe here is that you could see a suppression on the tumor volume of the cancer, and you can see that with our formulation most of the mice actually reacted. So it means that the particles, the nanovaccines, were able to stimulate the immune system in order to attack to produce more B and T cells in order to attack actually the cancer tumor. And you see here that no growth on the cancer, the melanoma cancer, while with the checkpoint inhibitor alone there was still most of the animals, the tumor was still active. So this is another beautiful application of how nanomedicines can be used for cancer therapy. But let me now go to the last part of my talk, where I'm going to show some examples also how the same systems can also be used for cardiovascular disease. So cardiovascular disease is still the disease that kills most in the world. It's the #1 disease, and after that it comes the cancer. So we had the same idea, why not apply the same kind of strategy in order to deliver drugs also to the myocardium, the infarcted myocardium. So why this is important? Because once there is a myocardial infarction, the heart tissue cannot regenerate back. So this is very important if we can somehow induce some stimulation or regeneration or reprogramming of the fibroblasts of the cells here in order that they can differentiate into cardiomyocytes. So this is what we've been doing. Another problem is that in order to do so we need to deliver a large amount of drugs, up to 7 or 9, a cocktail of drugs in order to do the job. So this is very difficult to formulate if we do it just in a normal drug formulation, but with these nanosystems we can do that. So that's what we've been doing in my lab. So we have modified the particles, the porous materials with targeting peptides. The particles look like this. The size is very small, less than 200 nanometers, and we have targeted them to the heart. So here what you can see, it's SPECT CT imaging, where we have injected the particles into the tail of the mice and the rat, and you can see that they accumulate very nicely in the heart region. So this is an infarcted heart. It is a disease heart. So what we had a porous silicon modified with a targeting peptide, and then we had radio-labeled them, and we have drug molecule that is a lead compound that is known to induce some tissue regeneration of the heart tissue regeneration. So what we see is that accumulation of that. So it worked really very nicely, and we were very happy to see that. So what we made sure that really the particles are the ones that are accumulating there. By TM imaging of the sectioning of the tissue, we saw that the particles really accumulate there without the target peptide. They don't accumulate with the target peptide, they accumulate, and they mainly accumulate in the medial part of the target tissue. So this is also very amazing, and it was very important to observe. But most importantly, you also can see here the properties. We were able to see that the lead compound that we are using is poorly water soluble, so it doesn't dissolve. That's why there is a 0 here. But when we put the 0 dissolution that we put inside of our nanosystems, you see that an increase of solubility, huge increase of solubility, enhancing of the solubility. And then we observed we looked to some of the proteins expression in the tissue after our formulation and treatment, and we look to the PERK, and PP38 proteins, and if these gray bars go down and these ones go up, it's a good sign, so it means that we are able to stimulate some of the tissue, so this is a great achievement. So currently what we've been doing, we've been modifying using different systems, co-loading with other model drugs that can also stimulate the differentiation of cardiac cells. We know that these cells, after injection, they are very stable at a physiological pH, but at the lower pH, cardiac pH, in the infarcted tissue, they start to dissolve. So this is also what we want, because then they can release specifically this cocktail of drugs exactly in the right place. So, and then we did a lot of bioactive studies, and here we analyzed the beta-catenin and SMAD3 expression. I'm not going into detail about these results, but basically what we saw is that if the cytoplasmatic beta-catenin goes up, and if we are able to inhibit the intracellular SMAD3 so that we prevent this translocation to the nucleus, then this is a very good result, and this also means that we are avoiding we are inducing stimulation of the cardiac cells and inducing their differentiation. We did this in fibroblasts, and now we are demonstrated this also in cardiomyocytes. And as we speak, as I'm showing this to you, we are also testing in vivo if we can achieve the similar results. So just to show you here what we have been doing, we've been a lot demonstrating here. Let me just go back shortly. So we have demonstrated that we can use this, and I don't have time to show you all the videos here, but we have demonstrated that these are really cardiomyocytes that if you don't see so well here, but they are beating cardiomyocytes, beating cells, and with our treatment we see that they behave and they beat much better after delivering the drugs there than all the other controls and all the other formulations. So this is a great achievement. And then to conclude, I would just like to tell you that these multi-composites are promising platforms to control multi-drug delivery, diagnostics, and therapy. So I'm almost in the end of my talk, and as a conclusion, I just very briefly, and sorry for the fast demonstration, but I just wanted to show you that multi-composites of nanoparticles, nanomedicines that can be very useful platforms for control multi-drug delivery, diagnostic, and therapy. Why? Because of these properties that I'm going to show you here. We can control physical chemical properties of these materials. We can make them very homogeneous distribution. They are biocompatible, meaning that they degrade and the degradation products are not toxic. We can load different kind of drugs and very high loading degrees. We can release these drugs in certain conditions very precisely. We can use them for combination therapy more than ever. Cancer, cardiovascular disease, we need to use a cocktail of drug molecules in order to do the job. So with nanosystems we can do it much easier. And we can also use them for theranostics and multifunctionality, such as therapy and bioimaging. And of course, when we want to translate this into clinic, we need to guarantee that we can have reproducibility, and that by using some technologies such as the microfluidics that we are using in our lab, we can do that. Very little variation batch to batch, so a very high batch to batch reproducibility, and also we are able to scale it up so we can get hundreds of up to almost 1 kilogram of these nanosystems per day. This is very useful when translating into the clinic for those tests on industrial applications. And of course, this is only possible with collaborators. All this work that I show you, collaborators from Finland and also from abroad, for many others. So I need to thank to them, and also to the large amount of foundations that we have. That my group has received funds, especially from Finland, Academy of Finland, and also the European Research Council and many other foundations, and obviously from the hard-working group. The 20 or more than 20 researchers that I have in my group that has helped to do all the job, and much more that I didn't show here today. And of course, thank you all of you for the attention. I don't have, unfortunately I cannot answer any of the questions that you might have, but if you are interested and if you have questions, please do email me here, and thank you very much for your attention, and thank you very much for the opportunity, and wish you all a great day over there. Thank you.